Okay, so let's talk about this. When you learned about this in class, or in the video, it was pretty straightforward. So here you can see this is a piece of DNA. It's just been broken up into lines because it was too long to put on my screen. But the top strand is running 5' down to 3', right? So this is a big, long piece of DNA that we just chopped up. And then the bottom strand is 3' to 5'. Okay, and normally you read DNA 5' to 3'. Just like we read left to right, we read DNA 5' to 3'. So this hemoglobin gene is running in a direction that you would normally see it run. So you can see here's our TATA box right here, and then the enzyme would come along and it would start copying right here, right? Or it actually starts at the TATA box, but this is where you're going to start reading the gene. And so you'd see the ATG or this TAC on the template strand, and you'd write AUG, CAC, and so on, all the way to the end of the gene, right? And then you'd have our polyadenylation signal right here where it would tell you to stop or termination. Okay, so that's all fine and dandy, but in this case, we are going to read the myosin gene. And if you notice, the myosin gene is on that bottom strand. So we actually need to read it 5' to 3'. So this is our 5' end, and this is our 3' end of this gene. It goes all the way along here to here. Okay, so in this case, we have to read it backwards. Now, one of the things we can do is just flip the whole darn thing upside down and read it the same way. So let's see if we can flip this over. We can. So I'm going to flip this over, and in this case, now we're looking at it the same way we looked at it before, from 5' to 3'. Whoops, here we go. 5' to 3', where the 5' end is right here, and the 3' end is right here. So again, you see ATG, so you would just write A, but a U instead, G, A, G, U, and so on, all the way to the end of the gene, okay? After that, you're going to be asked to process the DNA. So the bold parts we're considering exons, and the non-bold would be introns. So that's called DNA processing. We're going to take just the sections, let me highlight it in yellow. We're going to take just this section, and this section of our mRNA, and we're going to splice that together. So we got our 3 sections right there. And then the only other 2 things that have to happen in order for the DNA to be finished is we're going to add a 5' cap to one end, right? And then we're going to add our poly A tail to the other end, okay? And that all happens in the nucleus. That's where all processing happens. This will then leave the nucleus. So if we're looking at unprocessed DNA, we must be in the nucleus. Once it leaves the nucleus, then a ribosome is going to grab onto it, and it's going to read it and translate it into the amino acids, just like we learned in class.